I'm very excited to talk with you today. Um, just to start off with, back in March of 2020, when the COVID situation hit, it was a very interesting time for us at Fujifilm Irvine Scientific. We are a critical raw media supplier or raw component supplier for the production of different biotherapies and as well as vaccines. And one of the very interesting things back when I was talking with my team back in the middle of March, and we had sent some people home in order to try to be as safe as possible in the laboratory environment. But over the course of the next couple of weeks, we started to get a lot of requests from our large biopharmaceutical customers who are developing therapies and vaccines for COVID. And so I thought this was really interesting, especially for a lot of our entry stage people, because they came back to me and were saying, wow, we didn't realize how important cell culture media was to this whole process. So we've pretty much been in full work mode in the lab since about April of last year. And really proud of the team. But I do think that it really shed a lot of light on how important cell culture media is to the bioproduction process. So what I'd like to do today is talk with you about the cell culture environment, our approaches to cell culture media development, give a few case studies, in particular, T cells, CHO cells, and HEC293. For the multifunctional cell therapy approach, there are a number of different cell types that are used, of course, to make different treatment modalities. And then lastly, I'd like to talk a little bit just about the criticality of selecting a media supplier as well. So just to give a brief introduction, so Irvine Scientific has been around for over 50 years developing cell culture media for mammalian cell biology. Fujifilm acquired the company back in 2018, so a little bit over 2 years ago now, and has been providing a lot of support in bringing different technologies into the cell culture media space, which we've been able to leverage from a research and development standpoint. And our mission is to supply these types of products for the growth and handling of mammalian cells wherever we can contribute to the health and well-being of mankind. So as I mentioned just previously, we are fairly agnostic to cell types. So we're working with different immune cell types, the CHO, the HEC293 cells, MSCs. We also work with a variety of other cell types that aren't even mentioned here. But of course, all these different cell types are needed for these different treatment modalities, whether that be for adoptive cell therapies or antibody production for immune checkpoint blockades, the development of bispecific antibodies, cytokine therapies, cancer vaccines, oncolytic viral therapies, immune modulation, or more recently, the production of extracellular vesicles. So the ability to culture different cells and provide media for these cells is a big part of our research and development efforts. And this slide is essentially just showing that there are three major parts to the production process. One is, of course, the cell line, which is an extremely important aspect, selecting the right cells for your biotherapeutic production. But then the cell culture media is very important, and the process itself is extremely important as well, whether that be in small scale cultures, scaled up bioreactors, or very large scale production processes. And of course, ultimately, we're looking at yield and quality and the function of what is produced on the back end of this process. So the sum of all these inputs, it equals the level of performance. And this is our focus as we work with our customers and partners to develop cell culture media to fit their needs. Now, the cell culture media, of course, it consists of somewhere between 50 and 80 different components. So there is a lot of complexity to the media, and how we manipulate those different components, add, subtract, or change the ratios really has a very profound impact on the function and of the cells, as well as the yield and growth rates. On the bottom right-hand side of this diagram here, you can see the cell metabolism. This is just depicting a cell, and carbohydrates and glucose is a primary source of energy for the cell, of course. And as you go around the bottom side there, different types of lipids, of course, that then are used to make up the bilipid membrane of the cell, and also used for signaling purposes as well. And as you go around the bottom there, sometimes we're working with adhesion molecules, not always, of course, in suspension cultures. But then we are manipulating amino acid ratios, polyamines. Of course, the amino acids are used to produce the proteins, which are providing the structure and function for the cells. And then bringing in growth factors, hormones, and vitamins, and transition metals, and all of these different components mixed together create that cell culture media that supports the cell growth. So as far as the different types of approaches that we're currently using, so what we like to call as our approach is rational culture media design. And essentially what this means is that it's not one approach that works for cell culture media design. We do use a variety of different approaches. And so starting with the information that we have about that cell culture cell line, the cell culture process, and the end objectives from a functionality standpoint, we use slightly different approaches. So we are initially taking some of the knowledge that we have learned from that process and applying that. Potentially, we could also use some design of experiments when we're trying to drive very large experiments where we're testing many different components in parallel. Occasionally, we'll be using some high throughput screening techniques, which is more of a brute force type of a method to get through screening of lots of different compounds or lots of different media blends to get to a final result. We do go through a scalable process, so starting at a very small scale and moving up through to much larger scale, and oftentimes through bioreactor production. And we're also testing the characterization of all the different metabolites that are in the media during the course of the production process, so that we can see if there are things that we would like to change throughout the course of that process. We're also using more highly powered statistical techniques such as multivariate data analysis. In this case, we're looking for correlations between different components in the media, different concentrations of components, and the functional outcome. And so this multivariate data analysis in combination with our design of experiments does really provide a very powerful tool for understanding what is driving the cell culture process. And then much more recently, we've gotten into in silico media prediction. So this is the use of big data analytics and algorithms that are used to predict which media will perform most effectively with each different cell type. So this is a very iterative process. We are using, from a process knowledge standpoint, that experience, the data that we've generated, knowledge about and data from cell metabolism and biochemistry. We perform the experiments, and then after those experiments are performed, we're gathering that data, we go back, and then we apply some more of these this different rational culture media design approaches to develop the next part of the process. So we've gotten this down to a pretty controlled science at this point, and we can get through these tests relatively quickly. So in order to work with all these different cell types, and in order to gather the appropriate data, not only do we need to be able to culture the different cells in familiarity with those different cell types, we also need a variety of different techniques, different types of cell culture vessels and bioreactors and plates and flasks and bags, cell counting, flow cytometry, ELISAs, protein and DNA testing, quality analysis, the potency assays, media characterization testing, even through spent media analysis, and then high throughput media screening and nanoparticle analysis, which is utilized now for testing and characterizing exosomes and extracellular vesicles. So these techniques are also an extremely important part of the process. Now, from a scalable standpoint, as I mentioned, we do go from a very small scale and move up to a much larger scale. And of course, the smaller scale we're working at, the higher throughput. So you can see on the left-hand side there a biomech liquid handler, which can go through 96 well and 384 well plates very quickly. And we can culture cells at a very small scale. We then move up to more 10 milliliter scale through amber systems and smaller scale bioreactors. We do have small-scale perfusion systems as well, which is extremely useful because the perfusion systems and the media that's developed, perfusion media, is much different oftentimes than more static types of systems. So we want to be as close to our customers' manufacturing process as possible. And then scaling up to then, more what we consider medium scale or benchtop bioreactors that would go up to 3-liter scale. And then from there, it goes much higher. Our customers are working on much larger volumes than that. But once we get up to the 3-liter scale, it's generally predictive of what you're going to see at the full-scale manufacturing process. So getting into some of these case studies, starting off, we'll talk about a T-cell chemically defined media case study here. And just to start off with, this is an example of a media that does not perform at small scale the same way that it does at very large scale. And so this was a media that we have developed that really works at both medium scale bioreactor and larger scale bioreactor extremely well. And it performs okay at the static culture level, culture and plate. So oftentimes when we're working with customers who are testing this media, we're asking them to work with us and at that medium scale and larger scale level. So this is showing here going from static cultures up to the gas permeable bioreactors. We've been working primarily with a system called the G-Rex system at that medium scale, and then have also moved up into rock motion systems, stirred tank bioreactors, as well as hollow fiber bioreactors as well. So getting into some of the data here, you can see that this chemically defined media, when compared with some different competition media that's on the market, as well as more of a gold standard RPMI and 10% FBS formulation, that when we're looking at CD3 expansion, and this isn't a G-Rex system once again, and it's fresh PBMCs, and we're expanding T cells, we do see that there's a significant difference between the way that the chemically defined media works from an expansion standpoint compared to the RPMI and 10% FBS. And then if you look on the right-hand side there, looking at the viability of the cells, that there's not too much difference in viability. There you're talking about somewhere between 84 and about 94% from a viability perspective. So not too much significant difference from the viability standpoint. So then we get more into the functional testing of these cells and looking at, for example, the active state for T cells with CD25 or markers for more naive T cells with the CD62L, and then of course the exhaustion markers such as PD1. So when we're taking a look at the CDM chemically defined media compared to benchmark, compared to RPMI plus 10% FBS, that you can see there that over the course of a 10-day culture, there's no significant changes. The cells are in a resting state as you go from 0 to 10 days, looking at the CD25 marker there, and that they are very comparable media formulations when it comes to looking at this panel of markers for CD3 expansion. Okay, and then as we get into from a functional standpoint, so the immunomodulatory capabilities of the T cells, and does the media have any sort of impact on the T cell immunomodulatory capabilities? So in that top graph there, you can see that the ratio of CD4 to CD8 cells, approximately about a 2-to-1 ratio of CD4 to CD8. And that's at day 0. You go through the course of the culture, and you can see that the CDM media does hold up that ratio fairly well compared to the benchmark and the RPMI and 10% FBS. And so, which is a good sign. You're maintaining that culture in a standard ratio. And as you go to the bottom there and look at the expression of CD3, CD4, so the CD4 positive cells on the left-hand side, the CD8 panel on the right-hand side, and in this case, we're looking for the stimulation or the expression of interferon gamma, TNF alpha, interleukin 4, which of course are representative of the Th1 pro-inflammatory and Th2 states. And so you can see there through stimulation with streptococcal enterotoxin B, that you can get good stimulation, good immunomodulatory function of those T cells, whether you're testing the chemically defined media or the benchmark xeno-free media formulation. So very comparable to the benchmark, whether it be in the CD4 cells or in the CD8 cells. Okay, so in addition to the immunomodulatory function, we're also looking for the cytotoxicity and specifically at CD8, cytotoxic phenotype in T cells cultured in chemically defined media. And so you can see here the secretion or the expression of the CD107A for degranulation, perforin expression, or granzyme B, and basically through that similar type of stimulation through with SEB, you can get the stimulation of these cells and an increase in their cytotoxic phenotype. So they're functioning properly from an in vitro perspective. Okay, so then moving on to our CHO cell chemically defined media, which of course is used primarily to make monoclonal antibodies. We also work very closely with our customers, and different customers and their cell types can perform very differently. So we do want to make sure we are giving our customers lots of different options that they can test, and we do some of the testing in our laboratory as well. So this is broken into two different phases. This was a case study to improve growth titer and galactosylation in a fed batch process, utilizing CHO GS cells. So you can see here the phase 1, there's a media survey panel that we've developed with all the raw materials that go into these panels have been tested and approved from a quality standpoint for scalability and to be used in GMP manufacturing processes. So we have a media survey panel that can be used to test for growth and titer. Once we select the best growth in titer, we would move on to a phase 2 and optimize the media for the ability to have the appropriate galactosylation characteristics as well. So in this case, you can see in these growth curves, the top there is the viable cell density, the bottom is the viability. Basically, there are differences, which is a good thing with the media panel. We do see differences utilizing the media for CHO cells. And so you can see here some differences in viable cell density and the viability of the cells when we're looking at our panel, as well as some different media from suppliers that are currently out on the market. And so after testing that growth and the viability, we're also looking at the titer, the monoclonal antibody titer of the cells, and it's also good to see the differences here so that we can make some selections of the best media to start with from an optimization standpoint. So in this case, you can see here that the media panel 1 from a titer standpoint is really performing the best in comparison to the other medias that were tested. And then so in this case, we are also looking at the glycan profiles and utilizing analytical techniques to look for the glycan profiles. In this case, the customer was actually looking for a low mannose 5 levels. So we did move forward to after phase 1, moving into phase 2. You can see here the low mannose 5 levels for media 1 panel, as well as the expression of different glycans and galactosylation percentage as well in this table. So in this case, we're looking at all the performance of all the different media and making a selection to move forward into phase 2. So in phase 2 here, and this is looking at then a different supplementation or feeding panel here in the top right, where we are testing up to 30 different types of supplements and then looking at the galactosylation profile of those cells to determine if this is acceptable for the customer's needs. So you can see the differences there in the top. It's normalized to supplement 1, but we do have a number of different supplements that are giving higher galactosylation levels, which is something that the customer had desired. So in the bottom right-hand panel there, you can see that we've increased the galactosylation levels by 40 to 50%, utilizing supplement 6 in this example here. Okay, so then moving on to another cell type, which is the HEC293, and I had started off the presentation talking about how in the COVID situation, some of our customers are utilizing HEC293 for viral vector production. So this was an area of intense work for research and development over the course of the past year. And so we'll talk a little about the HEC293 CDM case study. And in this case, so the background, and we'll talk more specifically about gene therapy in this example. So this is for AAV production. And so AAV production is a leading platform, of course, for gene therapy. And but there are still some gaps in this process here. And some of those gaps are therapeutic doses. We also have gaps with the production costs being fairly high. And there's also this challenge where a majority of the AAV, the particles are consists of genome-free or empty capsids. And how do we increase that ratio of capsids that have genomes, viral genomes inside? And so you can see here the process that we were using. We were seeding 1 million cells, and then on day 2, where there would be 2 million cells, we would be transfecting the cells using a PEI transduction reagent, and then running the production for 72 hours. And so you see here that looking at the different supplements at different concentrations, that there are differences in the concentration of viral genomes per milliliter, as well as the ratio of capsid to genome and the production efficiency. So, it's a good sign from this development process. It's still a work in progress to try to increase our viral genome production efficiency here, but it just goes to show that from an approaches standpoint and method standpoint, that we have a process that can yield different results and promising results for our gene therapy customers. So then those are the case studies. Did want to talk a little bit just about the high quality from media development. It's an important thing to think about as a biopharmaceutical manufacturer, especially when you're starting out with your developing your process and your platform and selecting your media, just that you're thinking about some of these things and working with a partner who can help you from even a regulatory standpoint and a scalability standpoint. So the supplier's responsibility, from a media standpoint, really around the raw materials, understanding the quality of those raw materials, the safety of those raw materials, and then developing the media and making sure that the media is consistent and can be used in the production process. So a lot of what the media supplier can also do is, from a regulatory perspective, is provide the information that the regulatory agencies need. Because the media is so closely linked to the cells and the final product, the regulatory agencies are very interested in understanding what is going into the cell culture media and what is the control that the company has over that media and the raw materials themselves, to make sure that there is control over the process. We're not introducing any sort of adventitious agents, and that we're getting the same product each time. So a lot of this is driven by different regulations, and we see there like the ATMP and more of a European regulatory platform, but we are looking globally at the different regulatory agencies and what they need. Oftentimes developing drug master files to support the media, so that the regulatory agencies can utilize those drug master files and can reference them, and those can be used to support the filings. And then, as I mentioned a little bit earlier, going from a very small scale and up to the CGMP manufacturing, having a survey panel to start off with, where all of those components that are in the surveys have already been vetted, they've been qualified, and they can be then used in testing, and with the confidence that if you scale up those media, that they can be used for your production process. So that's a very important part. We do a lot of development, a lot of optimization of media. Some of that media development work, depending on what the customers would like, could be done in their lab if they don't want to transfer cell lines over. And we can provide them with different iterative panels of media that they can test and see what works best for them. We can help them analyze the data and then suggest another panel to them. There's also a complete service that we offer if the customer would like to send their cell line, and we could work directly with that, whether that be in California or a facility in Japan or in Europe, and can work with that and then develop a media as a complete service for a customer. So going from very smaller scale production, we have express media service, somewhere in the neighborhood of 1 to 18 kilograms of powder or up to 200 liters of liquid. It takes a shorter, 10 business days to produce that express media. And then once it gets up to the more very large scale, we're talking about, thousands of kilograms of powder or 10,000 liters of liquid in about 2 to 3 months. So the packaging is an important part of this also, whether that be the bottles, bags, multiple bags, liquids, powders, going all the way from that small scale, depending on what customer needs are, or up to larger scale when they're working in bioreactors or big vats and need different packaging configurations. So that's also an important part of the process. And then, as you're looking at the different media suppliers, just ensuring that there's an uninterrupted supply. Is there redundancy in the manufacturing? In this case, we have a plant in California, in Japan, and in Europe. And those are all of those production processes are utilizing the same raw materials, equipment, standard operating procedures. The quality systems are the same, and the way that we approach research and development and media development and optimization is very much the same as well. And that helps in case there's anything happens at one of the plants, and we can cover with one of our other plants in different areas of the world. So just lastly here, I mentioned that Irvine Scientific was acquired by Fujifilm a little over 2 years ago. Fujifilm has a big focus on the life sciences area and on healthcare and on bioproduction and contract manufacturing. So there have been a number of acquisitions of different companies over the past several years to put together different pieces that support cell biology. We're a media manufacturer. We do have stem cell developers such as Fujifilm Cellular Dynamics, developing some cell therapies. Some of those for the cancer cell therapy space. Fujifilm Diosynth, which is a contract manufacturing organization. Fujifilm Wako, which is a fine chemical and reagent supplier. So really trying to round out the different aspects of bioproduction and how we can support that and improve human health. So ultimately, at the end, we're looking to partner with customers, depending on what their needs are, what their process are. We want to work hand in hand from an early stage with them so that we can provide them with the best scientific expertise around media that we can, and that they end up with a high quality product that is functional and ready to treat people. So with that, I will stop and if there's any questions, be happy to take it. And thanks again to the organizers, and looking forward to the rest of the day. Thank you.
Thanks, Robert, for that great talk. I for one, always love to hear about new media. As you know, this is one of the big steps in going through trying to bring a cell therapy into the clinic. And we appreciate you sharing your work and the developments in your assays. I have a couple questions from the audience. The first is, how is the qualification of raw starting materials for cell therapy media different from that of monoclonal antibody therapies media?
Okay, thanks, Dr. Lamb. Yeah, good question. So, we are always taking a risk-based approach when we're looking at the qualification of raw materials, and the differences between the monoclonal antibody and cell therapy. So a lot of times with the cell therapy media, they can contain some different biological components of biological origin, and therefore that takes a different level of scrutiny. Most of the time we are looking at the regulatory requirements understanding what those regulatory requirements are in different areas of the world, and then applying those regulations to our selection of raw materials. So, for example, we're looking at the certificate of analysis, the certificate of origin, what type of adventitious agent testing has been performed on those components, and really taking that risk-based approach. So there are some differences between biological origin and non-biological origin. We're using different compendial methods for testing the raw materials as we select them from our vendors, and in making sure that we have really tight testing controls, as well as we're going through the paperwork exercise as well in the selection of those materials.
Have a second question that's related. What are the differences in CQAs considered for cell therapy, cell culture media, and monoclonal antibody therapies?
Okay, so yeah, so I mentioned a little bit about from the quality and safety aspect, the critical quality attributes that we'd be looking at. That be things like, the purity, of course, of the material, the sterility of the material. Are there any endotoxins that might be in that material, and understanding what the limits are that we're working with and that we can test to. So that's from sort of the safety side of things. There's also the critical quality attributes that would apply more to the functional aspects of the cell. So having the ability to perform those assays, maybe they're a cell-based assay or an analytical assay within our quality group, is an important part of the process as well. So we do want to make sure we have all of the all of the testing mechanisms in place, so that we can do the quality control testing here to meet the needs of the customer and the needs of the regulatory agencies.
Okay, I think that's all we have. Thank you so much for sharing that. We look forward to seeing your media working in different cell therapy programs and studies, and hopefully we'll bump into you live at a booth at a real live meeting someday.
Sounds great. Thanks, Dr. Lamb. And nice to meet you all on the teleconference today.